So our first lesson that we're going to look at today is the history of DNA, and there's a few key players that we need to know that are explicitly mentioned in the program of studies. Our starting point, however, is just going to be the general structure and function of DNA. So when we look at DNA, most of us, or talk about DNA, most of us are kind of like, yeah, we get what that is. That's like the molecule of heredity. That's, what carries our genes, and that's what transmits, information from parent to offspring in terms of our traits and our characteristics. And so, yeah, that's all true. And so we're going to base most of our study on the structure of DNA in eukaryotic organisms. And so just to refresh our memories with some of that terminology, eukaryotic organisms have cells that have a membrane-bound nucleus and membrane-bound organelles. So if we take a look at the location of DNA in a eukaryotic organism or eukaryotic cell, we can see that we have this nucleus here, we have this dark center here, which is the nucleolus. The nucleolus is responsible for production of ribosomes. We'll talk about that a little bit later. But we have this mass of coiled up genetic material, these long DNA strands called chromatin that are inside the nucleus of the eukaryotic cell. So when the cell is ready to replicate, then that chromatin, that thread-like formation of DNA will coil up. We call that supercoiling. It'll coil up into the actual chromosomal form, which is probably what we're most familiar with seeing as opposed to that chromatin. So in any case, when we look at DNA structure, most of us are familiar with this characteristic double helix. So DNA is a double-stranded molecule, as we can see here. Okay, when we kind of unwind that double helix, we can see that it has this side-like structure, like a ladder. Okay, so you have the sides of the ladder. That's called the sugar phosphate backbone. And then you have what we can kind of talk about as the rungs of the ladder or the steps of the ladder, and that's where we have our nitrogenous bases. And so all living things, regardless of whether it's a tree, an ant, a human being, they all have the same nitrogenous bases for DNA. And those four nitrogenous bases are guanine, cytosine, adenine, and thymine. So if we take a look at some of the main players in the history of DNA, there's a couple of different ones that are of significance. And so obviously thinking about genetic material and the discovery of inheritance and all of that goes back much earlier than 1944, but this is kind of our starting point for this unit. So in 1944, Avery and MacLeod and McCarty worked together to show that DNA was the transforming principle, and what that meant is that offspring were not genetically identical to parents because of DNA and because of that transformation of DNA that occurs during the production of gametes, eggs and sperm, and of course during fertilization. So then we also have one of the main players in our program of studies, a name that we do need to know, is Erwin Chargaff. And in 1944, he was the one that analyzed the composition of bases in DNA in a number of different organisms. And so what he kind of found is kind of that the fact that certain base pairs are the same in terms of their percentages in a strand of DNA. And then Rosalind Franklin, very important. Franklin worked with Maurice Wilkins, but she's the one that is specifically mentioned in our program of studies, not Wilkins. But in any case, her work was very significant because she worked using a process of X-ray diffraction to expose that helical structure of DNA. And so these photographs here that were done under X-ray diffraction or with X-ray diffraction showed that double helix structure. So her work and Wilkins' work was the first to kind of illuminate the actual structure of DNA, which showed that double helix as well as the phosphate groups on the outside. Incidentally, at the time when Franklin was working, it was unknown that exposures to X-ray was actually cancer causing, and she died of ovarian cancer, believed to be linked to exposure to the X-rays that she used in her work. So moving along then to the other individuals that are mentioned explicitly in our program of studies, we have Watson and we have Crick. So James Watson and Francis Crick basically took the work of Franklin and Wilkins, and they built on that and constructed that characteristic double helix structure that is still used today. And they did win a Nobel Peace Prize for their work in creating that actual physical model, that giant physical model of DNA. So they based their construction of this molecule on Franklin and Wilkins' work, and they were able to actually build that model. And so again, their model basically illuminated the fact that we have those 4 base pairs, the guanine, the cytosine, the adenine, and the thymine, that make up the rungs of those, the ladder. And then of course we have that DNA backbone, which makes up the side of those ladders, okay? And we'll talk about that a little bit more later. So just in terms of some general genetics terminology, genetic information, as we already said, in eukaryotic organisms is concentrated in the nucleus of the cell. So when we use the term chromosomes, these are those characteristic X-shaped bundles of genetic material like we see in our diagram here, and that is typically what we think about when we think of DNA. Keep in mind, however, that unless the cell is preparing to divide through mitosis or meiosis, that genetic material is actually like that bowl of noodles, okay? All of that, those chromosomes are actually unwound, and they actually are loose and kind of tangled up in the nucleus of those cells. So a couple other terms that are important when we talk about the different kinds of cells that we have as living organisms. We have somatic cells, which is the proper term for any body cell. So that would include things like a skin cell, a muscle cell, okay, anything like that. And then we have the gametic cells. And gametes is the proper term for sperm cells and egg cells, or collectively we call those the sex cells or the gametic cells. And so when we take a look at what we call the ploidy, and we'll talk about ploidy a little bit more in kind of the next section on mitosis and meiosis. But when we talk about ploidy, what we're talking about is how many chromosomes are there in these cells. So what's really important, and we're going to talk about a human here specifically, is in your body cells, there are 46 chromosomes. So if we look at our graphic here, and this is actually a karyogram, so it's where the chromosomes are taken and then paired together according to their characteristics. You can see that there are 23 pairs or 46 individual chromosomes, right? So we have pair 1, pair 2, pair 3, and so on, all the way down until we get to the sex chromosomes. So if you are a biological female, you're going to have 2 X chromosomes. If you're a biological male, you're going to have an X and a Y chromosome, okay? But that is considered the 23rd pair. So 23 chromosomes. 23 pairs of chromosomes. So that means that the ploidy of a body cell is going to be the full 46 chromosomes, okay? Now, when we talk about the gametes, so that would be the egg cells or the sperm cells, notice that they are only going to have 23 chromosomes. And the reason for that is because if you think about it, when egg and sperm unite, if we had 46 chromosomes in each of those gametes, we'd end up with what, 92 chromosomes in the offspring, which is not what the ploidy is supposed to be for a human being, right? We only want 46 chromosomes. So that's why 23 of the chromosomes come from the sperm, 23 of the chromosomes come from the egg, and then when that fertilization and conception happens, then we end up with our full complement of 46 chromosomes. So when we talk about chromosomes versus genes, there's a very, very important distinction. So when we talk about the DNA, we're talking about the full strand, right? All of the strands that are in that nucleus. When we talk about the chromosomes, we're talking about those pairs 1 to 23, where that genetic material, that chromatin, is supercoiled up into those characteristic chromosomal type structures. So when we talk about a gene, now we're talking about something different. And I really want to put pull your attention here to the graphic here. So when we're talking about a chromosome, of course, we're talking about this very characteristic bundle of genetic material. And then as we kind of unwind that chromosome or unravel it, now here where it's kind of pink, you can see that we now have that double helix. And when we talk about a gene, we are talking about a specific section. So notice it kind of is cropped out here, that pink part. So we are talking about a specific section of DNA that codes for a particular protein. Okay, so we have many, 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 many genes, okay, and those genes are specific segments of DNA that code for specific proteins. What kind of proteins? Maybe it's melanin for skin color or eye color. Maybe it's the gene for cystic fibrosis. Maybe it's the gene for looking at the sun and sneezing, okay? Every one of those segments of DNA that is called a gene means that it codes for a specific protein. So another term here is each gene occupies a certain location, or we say locus, on a specific chromosome. And so if we look at our little example here, you can see we have a chromosome that is responsible for example for the color of the flowers, okay? And then you can see we have two chromosomes here, and these chromosomes are what we call homologous because they have the same genes on the chromosome. So we have, you know, flower color here, and we have flower color here. Now, of course, one of these says white flowers and the other one says purple flowers, but it's still flower color. So the locus of flower color is the same on each of those chromosomes. Now, the color that the flower actually is different, okay, but the location of the gene for flower color is found on the same chromosome for that particular species of plant, okay? So when we talk about plural, so as opposed to singular, loci is plural, so that's all of the human genes along our 46 chromosomes, okay? So those are all the loci, and we call that the human genome. And that actually is in our program of study. So when we talk about the human genome, we're saying that we have 46 chromosomes. And we now know, because of the Human Genome Project, we now know exactly where the genes are that code for which proteins and in turn which particular traits. And that's going to be the same for all human beings. All human beings have the same human genome, meaning that the traits for every human, the loci are the same for every human on every chromosome. Now, obviously, we're all different because we have our own personal genome, right? Which means, do we make white flowers or do we make purple flowers? Okay, the genome is the same because the genes are on the same chromosomes for every human being that produce specific traits, and it's the version of those genes that is different, and that's our personal genome as opposed to our human genome. So when we look at the Human Genome Project, this was a very extensive project that involved like 18 plus countries, and it ran for about 13-ish years from 1990 to 2003. And really what it was a massive international collaboration to try and map the human genome, meaning we know we have 46 chromosomes, okay? Which genes are on which chromosome? Where are the chromosomes for eye color? Where are the chromosomes for height? Where are the chromosomes for Huntington's disease? Where are the chromosomes for eye color? Where are the chromosomes for breast cancer? Okay, where are the genes, I should say, for all of those different traits, okay? So 46 chromosomes, which chromosomes hold which genes, and then that's going to be true for all human beings. So about 120 genes or so code for height. So not just one gene, okay, it's lots of genes. We call that polygenic. So lots of genes contribute towards the expression of height. And obviously all human beings have different heights, right? But the whole point with the mapping of the genes is that those 120 genes that are responsible for dictating height in a human being are on the same loci of the same chromosomes. So it's not like, you know, you have all of your genes for height clustered on chromosome 1 and chromosome 2, and then your friend has their genes for height clustered on chromosome 20 and 21. Okay, it doesn't work that way. The genes that code for specific proteins and specific traits are on the same chromosomes across all human beings, and that's what we call the human genome. And we know that for a lot of different organisms now, okay? And this big research project, of course, was called the Human Genome Project. So just to simplify things a little bit, we can look at our fruit flies, or our Drosophila, which are a species of fruit fly very, very commonly used for genetic studies for a number of reasons. They don't have very many genes, they replicate very quickly, but this is the gene map for Drosophila. So you can see that we have the chromosome, and regardless of which individual fly we are looking at, the genes are in the same place. We have the gene for eye color, we have the gene for wing type, we have the gene, so eye color is here, wing type is here, body shape is here. So regardless of which fly you're talking about, their genes are going to they're going to be in the same place on the same chromosome. Now, which trait they end up expressing, of course, that is going to be different from individual fly to individual fly, but the location or the locus of the genes is the same for every fly. Okay, so what's the big deal about the Human Genome Project, and what does that allow us to do? There's a number of things that are useful when it comes to the benefits of the project. So first of all, now that we know where the genes are that are responsible for protein production and therefore different traits, we can actually do different things like compare individuals that have disease versus those who don't. We can diagnose genetic disorders. We can potentially correct genetic disorders, and we can use medications to help prevent and target specific gene expression. So there's a lot of things that we can do from a population genetics standpoint, as opposed to like just a medical standpoint. From a population study standpoint, we can actually compare groups of individuals to see how similar they are or how different they are by comparing their genomes, okay? And obviously, like we talked about, the human genome means on chromosome 1, we have these genes. On chromosome 2, we have these genes, etc. But however, your personal genome is what makes you distinct from all other human beings, okay? So yes, you have your genes for height on the same chromosomes everybody else does, but your genes for height have different combinations of alleles to give you different traits than other people when it comes to your height, okay? And we'll talk about that one a little bit later. So when we take a look at chromosomes, we do have a little bit of terminology that we need to know. And so when we look at eukaryotic chromosomes, these are held in place by proteins called centromeres, okay? So these little yellow kind of circles here are proteins that hold chromosomes together. Now, when we talk about chromosome studies, there's kind of two things that we talk about. So we talk about single-stranded chromosomes and we talk about double-stranded chromosomes, which visually you can kind of see, right? We have a single strand here, and of course we have a double-stranded chromosome here. But notice we don't say that this one on the right here is 2 chromosomes. It's not. It's still a chromosome, okay? A single chromosome. It's just that it's double-stranded instead of single-stranded, okay? So chromosomes are said to be made up of DNA and proteins, not just DNA alone. And of course, like we said, we have the single-stranded and the double-stranded. So just kind of keep that in mind as we move forward. When we talk about homologous chromosomes, that's a specific term as well. And what homologous chromosomes means is, think of that prefix homo means the same. And so homologous chromosomes means that they have the same genes in the same place, okay? So if we look at our little graphic here, you can think of, you get one of these chromosomes from your biological mother, and then you get one of them from your biological father, okay? So these chromosomes are homologous because they are the same size, the same shape-ish, and most importantly, the different regions, the different loci on the chromosomes code for the same gene, okay? So it's not like in a homologous pairing that this gene is for eye color, and then on this one's actually for toe length. It doesn't work like that. If these are homologous chromosomes, just like we saw with the purple and the white flower, this is for eye color, and this is also for eye color. It's just that one came from the biological mother, and one came from the biological father. but the genes are in the same place on the same chromosomes, and that's why it's called homologous, okay? So just another kind of terminology here. Chromatids are the single halves, okay? So we can see here that these are both double-stranded chromosomes, right? And so when we talk about a chromatid, that would be like one of these arms on the chromosome, okay? As we already said, the centromere is that protein in the middle that holds those chromatids together, okay? And then we also talked about chromatin, and so we talked about that a bit on the previous slide. So here's a micrograph of chromatin here, and then we can kind of see just the graphic here. So just keep in mind that unless the cell is ready to undergo mitosis or meiosis, which is cell division, we don't have these nicely little packaged chromosomes all lined up in our nuclei. We actually have this, think of it as a ball of yarn or a bowl of spaghetti. It's this kind of jumbled up, tangled up ball of like a single line of DNA, as opposed to it being coiled up into those chromosomal forms. So the last little piece for this section here is going forward, as I said, our focus definitely is on eukaryotic organisms, but in our program of studies, it does kind of ask that we understand the difference between eukaryotic and prokaryotic DNA. So here's the key. Prokaryotic organisms, which are mostly bacteria, they do not have a membrane-bound nucleus, okay? So eukaryotic organisms, that would be the kingdom Plantae, the kingdom Protista, the kingdom Animalia, most of the kingdom fungi. They are going to have that membrane-bound nucleus that holds their genetic material inside that nuclear envelope. They also have membrane-bound organelles like mitochondria and Golgi apparatus, etc. Now, prokaryotic organisms, primarily bacteria, you can see when we look at the graphic here that they do not have a nucleus. Instead, they have the nuclear material, they have that big strand of DNA just loose in the cytoplasm, okay? So it's not held in that nuclear envelope, in that nucleus. It is jumbled up in the in the cytoplasm of that specific cell. Now, there's also, moving forward, an important little term called plasmids. So the other thing that these bacterial cells have, and you can see here it says plasmid, is not only do they have this big strand of nucleoid or circular DNA, they also have these little teeny tiny little round, or sometimes almost like oval-like shaped little teeny tiny rings or ovals of DNA as well, called plasmid DNA. And that's going to become significant when we talk about genetic engineering and how we use bacterial cells to help us produce useful products like insulin, for example.